Dear viewers, greetings. In this video, we are going to see about the gram-positive bacilli, Bacillus cereus. First, general characteristics of Bacillus cereus. Bacillus cereus is a gram-positive rod-shaped bacilli arranged in form of chains, and it is motile in nature, and its size ranging from 3 micrometer to 9 micrometer, and it belongs to the family Bacilliaceae, and capsules present, endospores also present with central arrangement. The mode of respiration observed in the Bacillus cereus is aerobic respiration or facultative anaerobic respiration. The optimum temperature required for the cultivation of Bacillus cereus is 30 degrees Celsius to 40 degrees Celsius, and the optimum pH is 4.5 to 9.3. Regarding the habitat, Bacillus cereus is not considered as a normal flora of humans, but may be transiently colonize skin or the gastrointestinal or respiratory tract of the human beings. Bacillus cereus is isolated from soil, vegetables, milk, cereals, spices, fried rice, cooked poultry and meats, soups and desserts. Bacillus cereus is a saprophytic species of Bacillus species, and it widely lives in dead and decaying organic matters. Bacillus cereus is not easily killed by alcohol. They have been known to colonize distilled liquors and alcohol-soaked swabs and pads in numbers sufficient to cause infection. Regarding the discovery, an England microbiologist, Grace Franklin, isolated the Bacillus cereus from air in a cowshed along with her husband Percy Franklin in 1887. Disease transmission of Bacillus cereus. The primary mode of transmission of disease by Bacillus cereus is via the ingestion of Bacillus cereus contaminated food. Emetic type of food poisoning has been largely associated with the consumption of rice and pasta, while the diarrheal type is transmitted mostly by milk products, vegetables and meat. It forms spores and spreads very easily. Virulence factors of Bacillus cereus. The ability of the microorganisms to cause the disease is called as virulence. The term virulence was first coined by the scientist Louis Pasteur. Virulence factors are the factors which are responsible for causing an infection in human beings. These Bacillus cereus have the ability to produce two virulence factors. One is toxins, second one is enzymes. Enterotoxin is a major toxin produced by the Bacillus cereus, and it causes gastroenteritis, and it results in emetic form of disease that is caused by heat stable enterotoxin, diarrheal form of disease caused by heat level enterotoxins. Next, hemolysis and cytolysis are the activities of the enterotoxins. And next, the necrotoxin and cereulizin also produced by Bacillus cereus. And necrotic toxins are the heat level enterotoxin, and cereulizins are the potent humolysins named after the species Bacillus cereus. And the Bacillus cereus is also able to produce two enzymes. One is phospholipase C and the second one is collagenase. The phospholipase C acts as a potent lecithinase, and the collagenase acts on the collagen protein and degrades the collagen protein. Next, an incubation period of Bacillus cereus infection is 1 to 6 hours, and the infective dose required for causing an infection by the Bacillus cereus is 3000 to 9000 spores. Pathogenesis of Bacillus cereus. Bacillus cereus causes two types of food poisoning in humans, including diarrheal syndrome and emetic syndrome. Food poisoning results from its production of enterotoxin in the gastrointestinal tract, and the dosage of ingested Bacillus cereus spores leading to the diarrheal syndrome is 105 to 107 grams of ingested food and 105 to 108 grams of ingested food for emetic syndrome. Enterotoxins associated with diarrheal syndrome are unresistant to the acidic conditions of the stomach. However, the cerulite peptide toxin associated with emetic syndrome is more resistant to acidic conditions and remains active at 121 degrees Celsius. Although Bacillus cereus is commonly known to cause foodborne intoxications, it has been reported to cause local and systemic infections as an opportunistic pathogen, especially among the immunocompromised patients, newborns, and patients with surgical wounds. Finally, Bacillus cereus can also cause ocular infections. Three toxins have been implicated. They are necrotic toxin. It is a heat labile enterotoxin. Second one is cereulizin. Cereulizin is a potent hemolysin which is named after the species. And finally, the phospholipase C, a potent lecithinase. Bacillus cereus results in three clinical diseases in human beings. The first clinical disease is gastroenteritis, which includes emetic form of gastroenteritis and diarrheal form of gastroenteritis. The second clinical disease caused by the Bacillus cereus is ocular infection, and the third clinical disease caused by the Bacillus cereus is severe pulmonary disease in the lungs. The first clinical disease caused by the Bacillus cereus is gastroenteritis. Two forms of gastroenteritis are produced by Bacillus cereus. The first form of gastroenteritis is emetic form of gastroenteritis, and the second form of gastroenteritis is diarrheal form of gastroenteritis. And this table shows the difference between the emetic form and diarrheal form gastroenteritis. And first, the implicated food for the emetic form gastroenteritis is rice, and the implicated food for the diarrheal form gastroenteritis are meat and vegetables. And the mean incubation period for emetic form gastroenteritis is less than 6 hours and the mean is 2 hours, and the incubation period for diarrheal form is greater than 6 hours and the mean is 9 hours. Some of the symptoms of emetic form includes vomiting, nausea and abdominal cramps, and some of the symptoms of diarrheal form includes diarrhea, nausea, abdominal cramps, etc. And the duration for emetic form is 8 to 10 hours and the duration for the diarrheal form is 20 to 36 hours. And finally, the enterotoxin responsible for causing the emetic form of gastroenteritis is heat stable in nature, and the enterotoxin responsible for diarrheal form gastroenteritis is heat labile or heat sensitive in nature. The second clinical disease caused by the Bacillus cereus is ocular infection or eye infection. Bacillus cereus can cause ocular infections such as keratitis, endophthalmitis and panophthalmitis. A keratitis is an inflammation of the clear tissue on the cornea of the eye, and the second, endophthalmitis, is an infection of the tissues or fluids inside the eyeball, and finally panophthalmitis is an inflammation of all coats of the eyes. The third clinical disease caused by the Bacillus cereus is severe pulmonary diseases. Bacillus cereus can produce severe anthrax-like pulmonary diseases in an immunocompetent patients. Laboratory diagnosis of Bacillus cereus. The first diagnostic method is microscopic examination. Under gram staining, Bacillus cereus was observed as violet-colored gram-positive rods arranged in singles, pairs, or serpentine with square ends. In motility test, motile cells were observed, and in endospore staining, green color endospores with central arrangement was observed and red color vegetative cells were observed. This image shows the gram staining of Bacillus cereus. This is Bacillus cereus, which is observed as a gram positive rods. And this image shows the endospore staining of Bacillus cereus. Here, endospores were observed in green color and the vegetative cells were observed as red color. Colony morphology of Bacillus cereus on culture medium. In nutrient agar, Bacillus cereus was observed as large, feathery, spreading, dull, grey, granular, spreading colonies, and opaque with a rough matted surface and irregular perimeters. And in blood agar, Bacillus cereus shows beta hemolysis. And next in Meconic agar, white colored non-lactose fermenting colonies were observed. And in the selective medium, that is Bacillus cereus selective agar, Bacillus cereus was observed as peacock blue colored colonies. Next, biochemical test for Bacillus cereus identification. Catalase test positive, oxidase test negative, urease test negative, indole test positive, MR test or methyl red test negative, Voges score test or VP test positive, citrate utilization test positive, starch hydrolysis negative, casein hydrolysis positive, gelatin hydrolysis positive, nitrate reduction positive, arginine dehydrolase positive, esculin hydrolysis positive, lysis by gamma phage negative, and the lysis by gamma phage is positive for Bacillus anthracis and negative for Bacillus cereus. Finally, treatment and prevention. Oral rehydration or occasionally intravenous fluid and electrolyte replacement for patients with severe dehydration is indicated. Antibiotics are not indicated. Ocular infection is treated by using clindamycin, erythromycin, vancomycin, aminoglycosides and tetracycline. Proper cooking of food is one of the way for the prevention of Bacillus cereus infection, and refrigeration of food materials is not effective, but in some cases it also recommended to control the Bacillus cereus infection. Dear viewers, that's all about the Bacillus cereus. Thank you for the support. Thank you.